Hello and welcome everybody to this webinar about the preparation of cells and nuclei for genomic analysis. As the first point of the agenda for the webinar, I will explain the requirements for cell and nuclei preparation prior to genomic analysis. Next, I will introduce our workflow solutions to prepare cells and nuclei for genomic analysis, and in the last part, I will present some application genomic results from some studies. For the preparation of cells or nuclei prior to genomic analysis, there are several requirements that we need to keep in mind, especially for transcriptional analysis, and also depend on whether we want to perform bulk analysis or single cell and single nuclei analysis. For bulk analysis, cells need to be prepared only when the goal is to study a particular cell population, and nuclei need to be prepared as input for certain genomic analysis, such as ATAC-seq, which can be also performed at the single nuclei level. Therefore, to avoid transcriptional changes, it is important to work fast and apply gentle methods to extract the cells and nuclei. Then, for single cell or single nuclei analysis, there are, first of all, three main prerequisites in order to perform this type of analysis. The samples need to be debris-free, present high cell viability or intact nuclei, and of course, be a single cell or single nuclei suspension. And here again, it is required to process the samples as fast as possible with gentle methods to prevent changes in gene expression and to retrieve the different cell types present in the sample. This leads us directly to the workflow solutions that we can apply to efficiently prepare cells and nuclei for genomic analysis. These workflow solutions cover from the storage of solid tissues to dissociation and cleanup of cell suspensions that can come also from blood samples, as well as target cell isolation and cell analysis to ensure that the quality of the cell or nuclei suspensions is optimal prior to the genomic analysis. So let's start having a look to the first step of this workflow, the storage of tissue samples. Sometimes tissue samples need to be shipped to and from collaboration sites or be processed at different time points than the day of extraction. In these cases, a short-term storage of the tissues is a convenient solution, which is possible using the MACS tissue storage solution. We have validated this reagent on multiple tissues from murine and human origin for up to 48 hours at 2 to 8 degrees. It prevents the induction of apoptosis or necrosis of the tissues, keeping the cells with high viability over time, as you can see in this example of stored CD26 mouse tumors. The cellular composition of the tissues and the phenotype of the cells are also preserved, as we observed in this analysis of different TIL populations from the same stored tumors. And very important, the upregulation of cellular stress-related genes is prevented. This heat map represents default changes in gene expression levels of stress-related genes in stored mouse tumors referred to fresh tumors, showing that only minimal changes in expression of stress genes occurred on the stored cells. So once we are ready to process the tissues, then we proceed with the generation of single cell or single nuclei suspensions, depending on which of them we need for our genomic analysis. And here, gentleMACS technology is a perfect fit because it combines both automated mechanical disruption and enzymatic digestion of the tissues on the same platform. In this way, we are able to use soft mechanical forces to open up the tissues for the enzymes to react better, and also use less harsh enzymes and reduce incubation times. The mechanical disruption happens inside the gentleMACS tubes, and enzymatic digestion comes with the MACS tissue dissociation kits, and both are combined to work with the gentleMACS dissociators, where all the dissociation and incubation of the tissues can happen. So all in all, gentleMACS technology allows for the fast, gentle, and reproducible dissociation of tissues with the main goals of generating single cell or single nuclei suspensions, as well as preserving epitopes for downstream target cell isolation or analysis. And here I also want to highlight that these products are compatible products to the technology from 10x Genomics. The MACS tissue dissociation kits come along with optimized protocols to cover the dissociation of a wide diversity of fresh, murine, and human tissues, and also FFPE preserved tissues. The kits developed for the dissociation of fresh tissues are suitable to prepare cells for both bulk and single-cell DNA and RNA sequencing analysis. However, for FFPE preserved tissues, we have only validated the preparation of the cells for bulk DNA sequencing analysis at the moment. And for the preparation of nuclei for bulk or single nuclei analysis, the nuclei extraction buffer is the way to go for both fresh and frozen tissues. And I come to more details about this in my next slide. As I mentioned at the beginning, for some type of genomic analysis, it is required to prepare nuclei. And sometimes when working with certain tissues, such as snap frozen tissues, this is actually the only option, since no intact cells can be retrieved from such tissues. Therefore, we have developed a gentle method to extract nuclei from solid tissues based on the combination of mechanical disruption with the gentleMACS octodissociators and cell lysis using the nuclei extraction buffer. The protocol takes just 7 minutes, is performed at cold temperature, and up to 8 samples can be processed in parallel in a user-independent and automated way. We have validated this protocol with multiple tissues, including snap frozen and fresh mouse tissues, as well as OCT-embedded human tumor tissues. The graphs that you see on the left side show the nuclei genes obtained for each of these different tissues. And in the table, you see the RNA integrity number values obtained after RNA isolation from the extracted nuclei, indicating a very good RNA quality. But sometimes the cell suspensions contain unwanted material, such as aggregates, dead cells, debris, or erythrocytes, that need to be cleaned up to increase the yield of viable nucleated cells, which is particularly important prior to single-cell genomic analysis. To remove cell aggregates and clumps, results very convenient to filter the cell suspensions and also the nuclei suspensions using the MACS Smart strainers. They have a special design that prevents clogging and enables the fitting onto both 15 and 50 ml tubes, and have different mesh sizes are available for these filters. Dead cells can be efficiently removed using the dead cell removal kit, which consists on ready-to-use microbeads. It is a fast and straightforward solution to increase the frequency of viable cells that can be also performed automated using the AutoMACS Pro separator. The dot plots that are shown in the middle of the slide show an example of the removal of dead cells from a PBMC cell suspension that originally contained only 60% of viable cells, and after dead cell removal, it was possible to increase the viabilities to over 95%. Then, for cell suspensions that contain a lot of debris and red blood cells, we recommend to use our debris removal solution, which is a ready-to-use density gradient reagent that allows the fast removal of cell debris by centrifugation with full acceleration and full break, and our red blood cell lysis solution. In the dot plots on the right, you see how we apply both reagents to successfully enrich viable cells from a suspension of neural cells from adult mouse brain. So moving on with the steps of this workflow, after cleanup, we can isolate target cells using either magnetic cell separation or multiparameter cell sorting, or a combination of both. And I will give an example on when we can conveniently apply both later on. So if we go for magnetic cell separation, with MACS technology, we can isolate different cell types in just 3 easy steps. First, we incubate the cells with nano-sized magnetic microbeads coupled to antibodies that bind specific epitopes on the target cells. Second, we apply the cell suspension to a column placed within a magnet, and the labeled cells are retained in the column while the non-labeled cells will flow through. And in the final step, we can elute the labeled cells by removing the column from the magnet and gently flushing out the cells out of the column. These steps can be performed manually, but also fully automated using the AutoMACS Pro separator. And here also the products from MACS technology are fully compatible with those from 10x Genomics. By using MACS technology, we can nicely isolate specific target cells from either single cell suspensions after tissue dissociation, as well as from blood samples or PBMCs to get highly pure and viable target cells ready for genomic analysis. The MACS microbeads and the isolation kits guarantee full flexibility for positive and untouched isolations, and the straight from portfolio allows the convenient isolation of leukocyte populations directly from blood products. In addition to this, isolation of PBMCs from whole blood can be performed easily and automated with the whole blood PBMC isolation kit human and the AutoMACS Pro. The untouched isolation of PBMCs is done within only half an hour and yields pure PBMC fractions with minimal contamination of granulocytes or erythrocytes. On the bottom left, you can see the frequencies of granulocytes and erythrocytes detected in 3 PBMC fractions after isolation with density gradient centrifugation using FICOL or using the whole blood PBMC isolation kit. The presence of these cell contaminants was minimal when using the whole blood PBMC isolation kit, and the results were also more reproducible. Also, this method helps to minimize cellular stress responses that can occur during the isolation process. In the heat map on the right, we can see the full change in expression levels of a panel of stress-related genes in PBMCs isolated with FICOL compared to the FICOL-free method. And most genes were actually upregulated with the FICOL isolation, and this upregulation was even more prominent if we applied red blood cell lysis afterwards. Alternatively to magnetic cell separation, we can use flow cytometric cell sorting, which enables to isolate target cells based on the combination of multiple parameters. However, commonly used droplet-based sorters can damage the cells during the sorting process due to a high pressure or decompression within the fluidic system and electrical charges applied to the cells. So this type of sorting also releases aerosols to the ambient, which can represent a potential threat when we work with infectious samples. A more gentle alternative to droplet sorting is the MACSQuant Tyto cell sorter. And here, the sorting process takes place entirely in the MACSQuant Tyto cartridge, thanks to a unique microchip placed at the bottom of the cartridge. So no electrical charges or decompression are applied to the cells. The microchip contains a valve that opens once a target cell is recognized and leads it to the positive collection chamber. And the non-target cells are collected in the negative collection chamber and can be used for sequential sortings without compromising cell viability. Also, the cartridge is a single-use disposable, which eliminates the risk of sample contamination and carryover. And it is also a fully closed system, so it is possible to work with infectious material in a safe way. But before applying our samples to a cell sorter, we may consider to perform a pre-enrichment of the target cells especially if we are interested in cells present at very low frequencies, such as it can be the case of tumor infiltrating leukocytes. So this is the example that I wanted to show you. In the table, you can see the flow cytometry time per sample required to acquire a specific number of TIL populations from the bulk of the sample or after pre-enrichment of the cells, in this case using TIL microbits. So if we take a look at the CD4 positive T cells, for instance, for this particular sample, it took around an hour to acquire 5,000 cells. However, it took less than a minute to acquire the same amount of cells from the pre-enriched fraction. When we sort more than one sample, this time really adds up. And it can end up in having very long time periods for our sorting and analysis process, which is something that you want to avoid when performing gene expression analysis. But if you pre-enrich your samples, you will be able to acquire all the samples in much less time. And last but not least, it is very important to analyze the quality of the sample, including an accurate cell count and analysis of cell viability before the genomic analysis. And this can be easily done by flow cytometric analysis using the MACSQuant Analyzers. Accurate cell count is enabled with these instruments thanks to the volumetric pipetting feature that allows absolute cell counts in less than a minute without the need of counting bits. The cell count is integrated in every sample acquisition and can be performed fully automated. And now, in the last part of the webinar, I will present some exemplary results that were obtained in some studies. And yes, I will start with the first one. So in the first study, which actually was conducted together 10x Genomics, we aim to analyze the impact of sample preparation on genomic data quality. For this study, we use different syngeneic mouse tumors that we dissociated and filtered as a first step for cleanup. After filtration, we removed red blood cells, and afterwards we applied dead cell removal, either one time or 2 times, depending on the amount of dead cells in the cell suspensions. And after each cleanup step, we took an aliquot of the cells, and with all these aliquots, we performed a single cell analysis. From our single cell analysis, we evaluated different parameters to check the quality of the data that we got. And just to show you, especially one of these parameters, if we had a look at the cell recovery after all the cleanup steps, in this case, for the example of the colon tumors, for which we aim to recover 5,000 cells, you can see how well after all the cleanup steps, the cell recovery increased. So we were able to reach our goal of recover cells. And in addition to check cell recovery, we also analyzed the reproducibility of the method by analyzing just via visual inspection. So in here, we show the example of the breast tumors. And you see in different colors the different replicates, biological and technical replicates that we analyzed. And you can appreciate that there was a high degree of reproducibility across the samples after all the cleanup steps. And moreover, we were able to perfectly perform the analysis of tumor heterogeneity in the samples. And here you can also see the example of breast tumors, and in different colors, we depict the different populations that we detected in these samples, containing mainly tumor cells, but also we were able to nicely detect immune cells, fibroblasts, and other subpopulations in the sample. In our second study, we aim to perform a single-cell immune profiling of tumor-infiltrating T cells and B cells. In this case, we used human ovarian carcinoma samples that we stored overnight using our MACS tissue storage solution, and afterwards we dissociated and filtered. And because we stored the cells, we cryopreserved the cells, we also had to apply the cell removal. And we wanted to compare the resolution of the analysis of the T cells and B cells from the bulk sample, but also from isolated cells. So therefore, after that cell removal, we performed T cell and B cell isolation prior to the single cell analysis. In the dot plots on the bottom of this slide, on the left side, you see how we were able to nicely enrich the cell viabilities of the cells after thawing, where less than 30% of cells were viable to over 80% of viable cells. And from this cell suspension, we isolated either T cells or B cells, and the dot plots on the right show the nice enrichment on the T cell fraction. The single cell genomic analysis on the clonotypes of T cells and B cells revealed a clear increase in the resolution in the samples where we analyzed the isolated T cells and B cells. So in this case, the figures show the clone types that were detected for the T cells and B cells, either in the bulk of the sample or the isolated cells. And you can see a clear increase in the resolution of the analysis in the isolated fraction. And only here we were able to detect some of the clonotypes that otherwise were underrepresented or not even be able to detect it in the bulk of the sample. On a third study, we analyzed the single cell gene expression profiling of adult mouse neurons. In this case, we dissociated whole mouse adult brains, and after filtration, we applied two cleanup steps, debris removal and red blood cell lysis. And here we were also interested in analyzing the composition of the cells from the bulk of the sample or after neuron isolation, prior single cell analysis. The dot plots on the bottom of the slide again show you how the viabilities and the frequencies of neurons were before neuron isolation. So viabilities were quite good, but the presence of neurons was quite low. However, after a neuron isolation, we were able to nicely purify neuronal populations with also high viabilities. And when we performed the single cell genomic expression profiling of the isolated neurons, we were nicely able to identify subtype diversity and differentiate both populations of immature and mature neurons, which we were not really able to properly identify from the bulk of the sample. And in the last study that I want to show you, this was actually performed by Qui Nguyen from UCI. And in his study, Qui was interested in analyzing the effect of flow cytometric cell sorting in his target cells, which were murine epithelial cells from mammary gland. So first, we analyzed the expression of some stress genes, taking the panel that was published in this publication that you see on the left side. And he compared the expression of these stress-related genes on cells sorted either with a common droplet sorter or with a Tyto. And what he observed was that droplet sorting increased some stress responses in the cells compared to Tyto sorted cells. But in addition to this stress panel, he got more deeper into the analysis on analyzing the stress, the potential stress, and upregulation of other pathways related to these stress responses on cells. And here again, he detected that the cells isolated with MACSQuant Tyto had a lower expression of stress response genes compared to the droplet sorter. In addition to this, when he analyzed the number of genes that he could detect in the single cell expression analysis and the unique molecular identifiers, he saw that these numbers were higher in the cells sorted with MACSQuant Tyto compared to the cells sorted with droplet sorter. And moreover, he was also interested in analyzing the functionality of the cells after sorting. And for this, he performed a mammosphere formation assay and analyzed the generation of spheres after 7 days in culture. And here, the cells that had been sorted with MACSQuant Tyto generated higher amounts of spheres, meaning that the cells sorted with MACSQuant Tyto had higher functionalities compared to those sorted with droplet sorter. If you are interested in more details about these results, I would like to invite you to follow the link provided in this slide and watch the related webinar. Okay, and to sum up, I would like to remark that the path towards a reliable genomic analysis starts with the optimized preparation of your cells or nuclei suspensions. Gentle and fast methods for tissue dissociation, cleanup, and cell isolation help to get high-quality single-cell and single-nuclei suspensions. An efficient removal of cell clumps, dead cells, debris, and red blood cells increases cell viability and the yield of nucleated cells, which is particularly important for transcriptional analysis, and also the higher quality of the genomic analysis performed afterwards. And isolation of target cells leads us to increase the resolution of the analysis and also decrease cost, because we are only analyzing our cells of interest. And last but not least, flow cytometry-based QC allows the accurate cell counts and cell analysis to ensure that you reach the required standards prior to genomic analysis. And thank you very much for your attention.